Hi there. In this video we'll talk about cell signaling, which is the mechanism whereby information about the outside world enters into the cell and is met with an appropriate molecular response. In this video we'll talk specifically about different types of signaling pathways. We'll talk about some of the kinetics and specificity of signaling. We'll talk about the mechanism that the information is transferred, which the number one mechanism is phosphorylation of 3 amino acids, serine, threonine, and tyrosine. And then we'll talk about ways that cells amplify and otherwise modulate signals coming in from the outside world. This is fairly typical for the ways that biologists tend to depict these signaling pathways. You see a receptor here outside the plasma membrane, and then there's a lot of proteins on the inside with arrows being drawn, mostly indicating phosphorylation events, and usually finishing up with events here in the nucleus. Some protein will get phosphorylated, it will bind to nucleic acid, and it will stimulate the transcription of certain genes in response to the signal of the outside world, which in this case was the binding of the PDGF ligand. One thing biologists don't understand quite well is the apparent complexity that's depicted here, and we'll encounter that many times during our talks about signaling. This slide merely reiterates that cell signaling pathways are the way that cells communicate with each other, and that they're involved with just about every biological function known that cells undergo. Here we see sort of a generic schematic of the basic elements found in the signaling pathway. We have a receptor sitting in a lipid bilayer that binds a ligand which transmits a signal to various proteins. The proteins will eventually impinge on certain effector proteins, the dominant one being the gene regulatory proteins or the transcription factors, which are going to transcribe certain genes in response to the signal. Other things can happen. The effector proteins can be proteins themselves. This is a quicker response to a signaling event, which in this case could lead to altered metabolism, or you can also impinge on other proteins which are going to change the shape or the motion of the protein. Signaling pathway types can be classified based on the distance which they act over. The sort of the prototypical signaling event that people will think of occur over long distances, and that would be like hormone endocrine signaling, where a cell somewhere in the body releases a hormone. The hormone enters into the bloodstream, it distributes throughout the entire body, it will reach different target cells, bind to receptors, and so this can produce a systemic wide effect. Another long distance mechanism of signaling is nerve impulse. You have single nerve cells that can extend over a meter, and although the distance of the free diffusion of the ligand molecules very short distances, the signal will be input here into the neuron, and it can travel up to a meter away to the target cell. Short distance mechanisms include very important ones which are contact dependent, that the ligand is not free in solution, but it's bound to the signaling cell, and then the receptor is bound to the response cell. Nearly every cell participates in this kind of signaling, whether it's with other cells or the extracellular matrix. And then there's the short distance analog of endocrine signaling. This is called paracrine signaling. A ligand is released, but it doesn't enter into the bloodstream and go over all the body. It just enters into the extracellular fluid in the local environment and will go to nearby cells. I won't go into any of the details here. This is a table from your book, just shows some of the examples of very important molecules which you've heard about and the role they play in signaling. I'll just highlight insulin, adrenaline, testosterone, various growth factors, and neurotransmitters. The length of time a cell takes to respond to an external signal depends on its needs and the mechanism which is used. The predominant mechanism, which involves a DNA transcription response of new genes in response to a signaling event, can take minutes to hours to days because of all the time involved with RNA and protein production. This is perfectly acceptable for many things that the cell does, like development, the way when cells divide and grow organs and turn into organisms. Things, if they happen within a 24-hour period, is perfectly acceptable. If you have to respond quickly to an external stimulus, then you're going to want to bypass making RNA, bypass making protein, and simply alter the function of a protein that is present in the cytosol. In this manner, you can have a response in seconds or minutes. This slide makes a fairly simple point about signaling pathway connectivity, and that is that the pieces are inherently modular. This is demonstrated in this slide by the fact that the neurotransmitter acetylcholine, when it binds to the acetylcholine receptor in skeletal muscle, it will produce a responsive contraction. In the salivary gland, it will cause secretion of enzymes. In the heart muscle cell, it will trigger decreased rate and force of contraction, so that the same molecule binding to either the same or variety of acetylcholine receptors can produce completely different effects in different cells. It's all about the wiring inside the cell. Most signaling receptors sit on the surface of the cell, and large hydrophilic signaling molecules will bind to them. These hydrophilic signaling molecules, remember, are not able to cross large polar molecules or charged molecules are not able to cross the plasma membrane without being transported, but they can bind to a receptor and have a signal transmitted into the nucleus by the internal machinery. There's another class of signaling molecules which can be small and hydrophobic, like estrogen, which can bind to the estrogen receptor, which is a unique molecule in that it's a receptor and also a transcription factor. So we have a one-component signaling pathway where the hormone estrogen will bind to the estrogen receptor. The estrogen receptor will then transcribe genes on the DNA inside the nucleus. And in fact, that is what's depicted here, except not estrogen, but cortisol, which is a related hormone, binding to a receptor protein. Once it binds, it changes conformation. It gets translated into the nucleus and then stimulates transcription of a specific gene. Another example of a phenomenon is the signaling molecule nitric oxide. This is a gas which is produced in endothelial cells. It's formed from the breakdown of the amino acid arginine, and this small hydrophobic gas can rapidly diffuse into surrounding smooth muscle cells, binding a receptor protein, which then causes an effect for the smooth muscle cells to relax via the hydrolysis of GTP. This action of nitric oxide is the basis for what you may have seen on old TV shows where someone with a heart condition takes nitroglycerin tablets. They put them underneath their tongue when they're experiencing chest pains, and that will cause the blood vessels supplying blood to the heart to relax so that more blood can flow to the heart so that it doesn't have a myocardial infarction due to decreased amount of oxygen supply. On this slide, we see many of the different kinds of modules or pieces that can make up a signaling pathway. Again, the action starts out here where a hormone or a ligand binds a receptor. This activates it. A signal is going to be transmitted to successive proteins. In this case, a scaffolding protein holds these proteins in close contact. You can think of this as insulation or wiring that keeps the signal contained within the wire. So these are sort of relays in terms of transmission of the signal or the information. You can amplify the signal. You can integrate multiple inputs into 3 inputs here to could be required to produce a active conformation of this signaling protein, which can then go to another signaling protein in the cascade, and this protein can then spread it to multiple signaling intermediates so that more than one molecular consequences can occur because of the signaling event. You can anchor it to the cytoskeleton, hold it in fixed positions. You can block the responses by modulating proteins, and then again, generally the end result is a specific gene being transcribed in response to an environmental clue provided by the ligand. Okay, this will be a good place to pause, and we'll take it up with molecular mechanisms of information transfer. Thanks for listening.